Hello everyone, I'm Khyati Chandratre. I am a PhD student at University of Texas at Dallas. Today it's my pleasure to present my current research on 3D genome architecture and transcriptomic dysregulation in prostate cancer. So this particular project focuses on using a multi-omics approach in understanding the different structural dysregulation in metastatic prostate cancer. So as you know that eukaryotic interface nucleus, it forms, the chromatin is folded into a hierarchical organization where within the nucleus the chromosomes occupy chromosomal territories, which is then further segregated into two important compartments. Compartment A, which is transcriptionally active and it is closer in the nucleolus, and compartment B is the transcriptionally inactive compartment. Within these compartments, frequent interactions happen within the chromatin, which with the same physical, biochemical and functional properties. So because the chromatin interactions, they interact in such similar biochemical states, these interactions give rise to topologically associating domains which have CTCF and cohesion protein boundaries elements, which is enriched. So within these TADs, the chromatin further folds into regulatory loops, which brings the gene promoters closer to the enhancers, forming which is vital for regular gene expression and cell identity. So in healthy cells, this structure or organization is important in various nuclear functions such as gene expression control. So in cancer cells, this organization is disrupted, and because of this disruption, there is the genome undergoes miscompartmentalization, because of which certain genes which get access to enhancer elements which they usually do not interact with. So this causes activation a lot of oncogenic genes, which leads to tumor progression. So this structural dysregulation forms the basis of this research, where we essentially integrate a lot of different kinds of multi-omics data to understand the mechanisms which underlie the transcription dysregulation in metastatic prostate cancer. And for this, we utilize prostate cancer because prostate cancer disease is a compelling system in which the disease is characterized by various levels of enhancer activations or different levels of the disease, and there are various epigenomic factors or modifications also involved, which restructure the chromatin architecture in a way which causes various levels of regulatory changes on the transcriptome level. So because the prostate cancer is enhancer and epigenetic driven, it forms an ideal system in which the 3D the mechanistic basis of 3D genome architectural alterations could be studied. So in this particular study, we have generated a lot of we integrate 3D genome along with epigenome data and transcriptomic data to really understand how 3D genome alterations influence the transcriptional dysregulation in prostate cancer, and also how these changes further happen when the disease turns into metastasis. So to understand this, we use multi-omics data from 4 cell lines: normal cell line, which is RWP, and metastatic prostate cancer cell line, which is LNCAP, derived from lymph node metastasis; VCAP, which is derived from vertebrate metastasis; DU145, which is derived from brain metastasis. And we then generate the 3D genome interaction data is mainly focused for RNA polymerase, which modulates or which controls the gene regulation, and cohesin, which is a closed ringed complex which forms the topologically associating domain. So this cohesin ring structure has a specific subunit which is called Claisen subunit, and this subunit forms the it forms a closed ring cohesion structure, which kind of helps in forming a topologically associating domain along with the CTCF. So it essentially helps in forming an insulation forming a TAD or a domain within which the regulatory loops are modulated by RNA polymerase. So the first aim for this particular study is we want to quantify the relationship between the structural the looping patterns mediated by cohesin and RNA polymerase, which orchestrates transcription regulation in the 3D genome for in metastatic prostate cancer. So the aim is to quantify the relationship between RNA pol2 and cohesin mediated interaction. So for that we get the enrichment of RNA pol2 loops with cohesin enrichment in one peak, both of the anchor peaks, or none of the anchor peaks, and we found that more than 90% of these loops have enrichment of cohesin in one or both of the anchor peaks. So in the reverse scenario, we check cohesin loops with the enrichment of RNA pol2, and interestingly we find that only 50% of these loops have enrichment of RNA pol2 in one or both of the anchor peaks. So this asymmetry suggests that most of the RNA pol2 loops are mediated by cohesins. So then we further look into the enrichment of different regulatory loops mediated by RNA pol2 and check if there is any role of cohesin complexes in modulating or mediating those regulatory loops. So we see that a majority of RNA pol2 associated enhancer promoter loops are facilitated by cohesin. So in this poster we clearly show one example of VCAP cell line. So in VCAP cell line, which is an aggressive metastatic cell line, prostate cancer metastatic prostate cell line, wherein there is co-amplification of androgen receptor locus, that is AR, and its enhancer element. So in this particular setting, from previous studies, it's been shown that this AR locus amplification and its 700 kb upstream enhancer co-amplification leads to the progression of PSA, which is prostate specific antigen, and progresses the tumor in the prostate cancer. So here we integrate the chromatin interactions along with different sets of multi-omics data like ChIP-seq to get the binding sites of the transcription factors, CTCF, and then also gene expression to clearly show that the AR locus is amplified and along with the enhancer. So because of this co-amplification, the AR gene is co-expressed and it causes like an expansion of chromatin interactions, which is modulated by cohesin and RNA polymerase. So essentially we link with this particular study, we show that cohesin specifically plays an important role in forming the strong long range enhancer promoter interactions in the VCAP cell, which aids in the over expression of AR gene in prostate cancer. Then we further look into this was the part of the first aim. Second aim is to assess the stability of cohesin loops following a BET degrader treatment. So BET is DBET1 is a BET protein degrader, which specifically targets BRD4 protein. BRD4, it binds to acetylated enhancer regions, and then it modulates the expression of oncogenes by recruiting RNA pol2. So because so BRD4 is important for enhancer activity, and this enhancer promoter loop is facilitated by cohesin. So that's why cohesin is essential for enhancer connectivity. So that's the reason why we sought to understand what's the effect of DBET1 treatment on the VCAP cells, because DBET1 targets BRD4 and essentially it selectively degrades it by ubiquitination and proteasome with a proteasome activity. So what we found is that DBET1 VCAP cells treated after DBET1 treatment did not show any specific changes on the strength of the loop or the length of the loop, and there was no major changes in the looping patterns in the specific dbet target gene, which is MYC and AR androgen receptor. And because such super enhancer and gene promoter interactions, they happen within insulated neighborhoods, we also check the we also check the differences in the strength of the longest cohesion loops, which are between a super enhancer and its interacting gene, closest active gene, with a 50 kb window within the enhancer promoter interaction. And we get the strongest cohesion interaction and compare it with DMSO, which is the vehicle control, and dbet1 treated VCAP cells. And we found that from 50 kb window, from 0 kb until 500 kb on either sides of the super enhancer and the promoter loop, we see that there is no significant changes in the strength of the interactions. So what this essentially tells us is that the cohesion loops are so stable that even after treatment of BRD4 or BET protein inhibitor like dbet1, it affects the transcription of the gene, but it does not affect the stability of the cohesion loop. So cohesion loops are stable and they are refractory to any transcriptional inhibition perturbations. So then we move on to the third aim of the study, where we characterize the RNA pol 2 associated promoter loop interactions and co-regulated gene expression. So in this part of the aim, we understand the landscape of RNA pol 2 promoter interaction and link it with the gene expression. And we show that in all the 4 prostate cancer and the normal cell line, we see the phenomena that as you increase the strength of the promoter interaction, the gene expression increases. So essentially, as the loop gets stronger, there is higher co-expression, which suggests there is a coordination and synergy in the transcription control, which is also observed. We then checked for different valent interactions, from bivalent to multivalent interactions, and we essentially saw that there were a lot of strong interactions from 2 promoters until 11 promoters, wherein the frequency decreased as the valency increased. So there were more promoters with there were less a number of promoters were interacting highly compared to more promoters interacting frequently. So the strongest interaction was found with multiple promoter was found in VCAP cell. It was also found in other cells, but here as a representative, I'm showing it only for VCAP, where we see that the histone gene cluster had like a strong RNA pol 2 interaction, suggesting involvement of protein promoter interaction in coordinated gene expression for these genes, because essentially histone genes is regulation is important in early cell S to G1 cell cycle phase, where histone gene stoichiometric production of histone gene is really important to maintain for the wrapping of the newly synthesized DNA chromatin assembly in the DNA replication process. And then we further use this gene pairs connected by a promoter interactions and check whether this co-expression or the correlation is also found in the tumor sample. So we use the West Coast Dream Team patient cohort for using this molecular subtypes for adenocarcinoma, double negative, and the neuroendocrine, which provide transcriptional landscape similar to LNCAP and VCAP AR sensitive cell lines. And we do 2 comparisons. First comparison is between AR positive and negative samples, where we hypothesize that it should have higher correlation in AR positive samples compared to negative samples, and that's the exact expectation, and that's what we see through the correlation plot, that we have higher correlation in AR positive samples. And then we also do another comparison where we check the gene pairs which are connected via a stronger loop compared to a weak loop in AR positive samples. So here also we clearly see that the stronger loops have higher correlation compared to the weaker loop. So what this essentially tells us is that we have RNA pol 2 interaction data, and we analyze it alongside the correlated gene expressions, and then we use those correlated gene pairs to check the co-expression in the tumor sample. So what this essentially tells us is that this coordination of gene expression is not a cell line specific phenomena, but it is more generalized mechanism which is also found in tumor samples. So then we further move to the AIM4, where we examine the influence of DNA methylation and enhancer proximity in gene regulation. So in this AIM, what we want to understand is, thus far throughout the study, we understand that cohesin mediates a strong, long-ranged interaction between an enhancer and promoter. So what's the mechanistic basis when the enhancer is very close to the gene promoter, or it's very proximal, or just like right adjacent to the promoter? How is that modulated? So to understand this, we use GST-Pi1 gene. This gene is highly expressed in normal cells, and because it's responsible for cellular processes like fighting against it reduces the oxidative stress, and it is silenced in the cancer cells through promoter hypermethylation. So we understand this mechanism using GST-Pi1 gene, and to examine this, we use cell line RWP, where this gene is expressed, and 3 metastatic prostate cancer cell lines, LNCAP, VCAP, and D145, and patient-derived xenograft sample 78 and 77, which is lymph and bone metastasis sample, to understand what the differences in this mechanism where this gene is not expressed. So what we essentially found out that in the cells where normal cells where this gene is expressed, there is enhancer. We use a lot of integration of H3K27AC, RNA Pol2, ChIP-seq, DNA methylation, and RNA-seq to understand this mechanism. And what we found was, in normal cells where the gene is expressed, then there is enhancer activity. The promoter is not methylated, and the Pol2 is right at the transcription start site. But in contrast to that, we see that in LNCAP and VCAP, the promoter is heavily methylated, due to which this enhancer activity is shut down and it is disconnected from the promoter of the gene, redirecting Pol2 away from the TSS, and the gene is not expressed. Further interestingly, in D145, there is a focal decrease in this methylation at the promoter, which brings back the enhancer activity, redirects to Pol2 and at the TSS, and the gene is expressed, but it's in the expression is low. And this the same phenomena of enhancer disconnectivity from the promoter is also seen in the PDX samples. So what we understand from this is that in cases where the enhancer is disconnected in the promoter, or like along the promoter, enhancer is disconnected from the promoter due to methylation, and an upstream cryptic enhancer is activated, which is still not able to activate the gene expression. So essentially, it's like the enhancer is there, but it's not decommissioned. So what we understand from this AIM is that in long range enhancer promoter interaction, the interaction is mediated by cohesin, whereas when the enhancer is right next to the promoter, very proximal to the promoter, DNA methylation can tune the regulation of the gene expression. So with that, the final conclusion of this particular study is that cohesins are key determinants of RNA Pol2 associated chromatin looping. Cohesin loops are very stable and refractory to any transcriptional perturbation. We discover promoter-promoter interaction phenomena and its co-regulation of gene expression, and validate in the patient tumor samples. And finally, long-range interactions are mediated, enhancer promoter interactions are mediated by cohesins, whereas enhancer, which is right next to a gene promoter, it can be tuned via DNA methylation. So with this, that's all I had to present today. I thank you for this opportunity by ICB ECCB 2025 conference for giving me a chance to present my research, and thank you so much.